Great. So Alice, wonderful. Thank you for joining. And Alice, I'm just going to read your bio, is a freelance designer and a recent graduate of Toronto Metropolitan University's Bachelor of Interior Design program. She previously completed an honors BA from the University of Toronto in International Studies in Culture and Communications with minors in Sociology and Media Studies. Working together, we had the pleasure of working on projects related to COVID-19, and especially from the research side, the design side, and eventually worked on a project through an international project titled Interior Archipelago, which is a whole other website if you're interested in viewing that one. And the project's findings were presented at digital and physical exhibitions, conferences, and published in a book titled Interiors in the Era of COVID-19. Their research sparked an interest to share and further their investigation into the impact of the interior and its layers on human health. So those were some of the things that came up in the chapter that we worked on. So Alice is a multidisciplinary designer with interests that include research-based design, placemaking, and emotive architecture, and designing for health and well-being. So with that, Alice, I will hand things over to you. Thank you.
Thank you, Lois, for the generous intro. Good afternoon, everyone. I'm Alice. So having worked under Lois as an RA and a recent grad of TMU's Interior Design program, I'm really grateful to be able to connect with the school again and excited to see everything come together, and now kicking off the second session of this weekend's Healthy Interior Design Symposium. The recommended 6-foot distance around individuals marked by bubbles, signage, and markings on floor and ground surfaces during COVID-19 did not convey how far infectious droplets could really travel through coughing, sneezing, talking, and breathing. This session focuses on understanding the difference in particle sizes and units of measurement when viral droplets become aerosolized, and how visualization can inform and guide interior designers to integrate these units of measurements in developing solutions for healthy interiors. Two speakers contributing to the session are Dr. Kimberly A. Prather, Distinguished Chair in Atmospheric Chemistry at the University of California, San Diego, and Mr. Gabriele Gianordelli, Assistant Editor at the New York Times. I'm pleased to welcome Kimberly A. Prather, PhD, Distinguished Professor, Distinguished Chair in Atmospheric Chemistry at UCSD, Scripps Institution of Oceanography, Department of Chemistry and Biochemistry. Her research focuses on understanding the sources, composition, and reactivity of atmospheric aerosols and their impact on climate and health. As a founding director of the NSF Center for Aerosol Impacts on Chemistry of the Environment, she leads a team studying the ocean atmosphere system, particularly the influence of marine phytoplankton, bacteria, and viruses on atmospheric chemistry and climate. During the COVID-19 pandemic, Professor Prather advocated for recognizing aerosol transmission to control the virus's spread, sharing her insights through media and briefings with health leaders. Prather's contributions have earned her elected memberships in prestigious societies, including the American Philosophical Society, American Academy of Arts and Sciences, National Academy of Engineering, and National Academy of Sciences, and fellowships with the American Association for the Advancement of Sciences and the American Geophysical Union. Thank you for joining us today, and I'll hand the screen over to you, Dr. Prather.
Okay, well, thank you for that introduction, and also thank you for the invitation to be here today. I'm already learning a lot, and I feel like I know many of you. I'm trying to see, hold on, I can't walk and chew gum at the same time. Sorry, let me just make sure. I almost forgot to share my screen, which is probably important. Here we go. Okay, so now how do I hide these? Okay, those are, I gotta hide, I can't stand these floating controls. Okay, I think I'm almost there. All right, and then I just have to do this, and I think I'm home free. Okay, so can you see everything? I just want to make sure that I still see like the band of speakers to the side. Is it clear for you? Can you see the slides?
We also see the banners, I think, on top of your slides.
Okay, let me see if I can get rid of it. Oh man, I thought I was ready to do this. Let me just I'll just slide them down. And it puts it along the bottom. Okay, how about that? Does that work?
Yeah, that's better.
Okay, thank you. All right, well, again, thank you for the introduction, and thank you for the opportunity to be here. I was just thinking about the broad range of people that I've spoken to. I've spoke to a dentist convention, medical doctors, healthcare, and now, designers. So I, as you'll hear today, I love interdisciplinary work. I think that's the only way we can solve a lot of the problems that we face. And I was excited when I got Lois's invitation to speak today. And I'm kind of stepping back and just sort of thinking about the big picture again, and the way that, sort of what we've learned through the pandemic, what we knew before. And so I will sort of also share some of the frustrations along the way that I'm in line with Dr. Fissman. But he was one medical doctor who gave me hope, because early on he flipped and went from this, what we'll talk about as droplets, to aerosols. And so he gave me a lot of hope early on, and we have had many conversations propping each other up over the last 4 years or so. So just a little bit of background on me, and then maybe it'll make sense on how I got involved in this pandemic. But I am the director of a National Science Foundation Center, which focuses on aerosols. And it is a large interdisciplinary center, involves 12 different institutions. I've been the director for now 13 years. And it, as I say, basically focuses on aerosols, but it focuses on aerosols that come from the ocean. And I'll give a little bit up front. You might think, how did someone who studies aerosols coming from the ocean start to study aerosols from infectious disease? And hopefully by the end, I will have shared with you how I got to this point. But again, I think just running this center has really opened my eyes to the power, as we've talked about today, of having people from different fields that work together. Okay, so hold on. All right, so this is sort of my background. I have studied aerosols and how they circle the globe. You can release aerosols on, dust on one side of the planet can travel all the way around the Earth in about 2 weeks. And so I've studied their effects mostly on climate and how they affect clouds and weather. And as I say, you can see here that, we share the air no matter where we are. The air from one continent can go to another continent, not necessarily attached to a person. It can just get there, as I say, flowing through, in this case, the jet stream. And so that was sort of the background and what I came into this problem with, or into this particular, the issues with. And thinking about bioaerosols has become a really much bigger part of the research that we were interested in. So just to give you a little flavor, I will talk quite a bit about the size, and sort of because the size controls their behavior in many ways, as I'll explain in a second. But basically, aerosols span a pretty broad range of sizes, all the way from a nanometer, sort of down at the molecular scale. RNA and DNA and proteins. Viruses come next, bacteria are slightly bigger, and then you get the really big stuff, which is where I started my career, as I mentioned, looking at sea spray aerosols and dust in the atmosphere and their influence on climate and health. And so I've sort of gone backwards and moving down the scale and developing tools to look at smaller and smaller things that become suspended in the air. One punchline is sort of the smaller they are, the more likely they are to remain suspended in the air. Heavier things, big droplets, we call them cannonballs, will fall to the earth pretty quickly, as I'll talk about. So our focus has been on aerosols, which by definition refer to a stable suspension of aerosol particles, little bits, microscopic bits of things, including pollution. These little tiny pieces can stay, or particles can stay in the air for hours. So the challenge that we faced in CASE early on was to look at sort of what gets out of the ocean. And we were very interested. People thought, oh, salt, of course, right? The ocean's mostly salt. That's all you'll see. But there's been studies done clear back to the 50s that show that aerosolization is a really complex process, and the ocean contains all signs of life, including viruses and bacteria, proteins, sort of all of these things are enriched, and they get heavily enriched in this sort of thin layer of oil that sits on the surface called the sea surface microlayer. And it's just teeming with life. And so it is basically composed of things that don't want to be in the water. They're what we call hydrophobic, water hating. And so they tend to be released into the air. And so I had a real interest in sort of understanding this, started working with oceanographers, I'll show in a second, to really understand what gets out of the ocean. Is it just salt, or do we actually start to see these other things making it into the air. And so this, I've fallen in love with bubbles, and I've given whole lectures just on bubbles. But basically aerosolization from the ocean is one of my favorite, it's a hobby and my job. I get paid to look at these cool bubbles. But the point is it took us 3 years to sort of reproduce the ocean in the lab with real seawater, with real biology, and the right bubbles. The bubbles, by that I mean the bubbles that look just like the open ocean. And so you can see this is a breaking wave in one of our big simulators that we have in the lab. And so we've been really interested in which things get out of the ocean. There's 2 processes that happen on the surface of the ocean. Hint, they also happen on the surface of your lungs, where you have this film drop production, where basically that film, that thin hydrophobic film I mentioned, builds up, bursts, and then you can see the spray coming off. Then it's followed by jet drops coming up out of the middle, and bigger things come up out of there. And so when you get this process right, it is, you start to see structures that nobody really anticipated. We can see bacteria with being invaded by phage. We can see droplets with little bacteria inside. We even see little micellar structures that almost look like cells. And some people think that this may be one thing that led to or helped lead to sort of origin of life questions. But the point for this particular talk is that I have focused a lot of my research on sort of understanding what gets out. And we've shown in our research that only certain things get out, only certain types of bacteria, only certain types of viruses actually make it into the air. It's a very selective process. And the only way we know that is because we got the bubbles right by working with oceanographers. So that's sort of where I came in, just to give you a little flavor on my background. But now I want to sort of give, sort of the history of my involvement with many other people we call Team Airborne. And Dr. Fissman has been part of that. Shelley's been part of that. But thinking about the airborne transmission specifically to SARS-CoV-2. So initially, again, knowing my background, I was asked by a reporter, do I think that SARS-CoV-2 could go further than 6 feet? We all know, and it's in the description for this session, there's these bubbles that tell you stay 6 feet away and somehow you are magically protected. Well, when I was asked this question, this picture, the video that I showed at the beginning, immediately popped into my head, and I honestly laughed. And as they say, the rest is kind of history. I basically, they were surprised when I laughed, because I've studied things that have gone tens of thousands of miles. So yes, I think they can go further than 6 feet. And this led me to think more deeply about what was leading to this spread. Why, as an aerosol scientist, was I not surprised that this aerosol was making its way around the globe? And so I worked with Science magazine to write a perspective, and I worked with an infectious disease doctor. So it wasn't just an aerosol scientist lecturing the medical community. We both came out with sort of, this is how SARS-CoV-2 has sort of snuck its way around the globe. This is why universal masking is so important. And a big way that this particular virus has been tricky, and maybe many others, but this is the one where it has been really obvious, is if you remember early on, the spread was largely by people who weren't coughing and sneezing. So they weren't producing big boulders and droplets. They were actually producing lots of aerosols just by talking, or as Shelley talked about, just by singing. And so, we really hammered home the point of, let's connect the dots. This being spread by people who sometimes don't even know they're sick at this point, producing lots and lots of aerosols. And so the pandemic messaging was and David talked about this already, this tweet is indeed still up by WHO. It's very frustrating to many of us. They initially in February said it was airborne, and then they immediately took it back, and then sort of started basically avoiding all use of the word airborne from that point forward. And so because people were so focused on big stuff, the boulders, that's where these distances came in. That's where the 6 feet really showed, mattered. But what I will say is, both aerosols and droplets, if you're right up close to someone, the only thing that can protect you in that situation is a really good fitting mask. And distance helps you for both aerosols and droplets. And so, but a focus at the very beginning, if you remember, was, wiping down your groceries when they would come in, cleaning surfaces. And then, as I say, maintaining this distance of 6 feet. The other thing was just the language, again, was really there was it appeared to be a concerted effort to avoid the use of the word airborne. But as an article in Wired, it was a really good article, came out, said, they say it isn't airborne, but it's definitely borne by air, right? And so again, just from a physical perspective, it was absolutely stunning to be involved in sort of watching this wordsmithing and denial of the airborne transmission of this virus. And so in addition to this perspective was downloaded like 1.6 million times, basically, so I know people read it, but for some reason it just, as I'll talk about, we're still having this discussion. We also wrote a letter. This was after National Academies meeting where we discussed there's this magical 5 microns that people thought, like below 5 microns, maybe it'll float, but there aren't many of those. But above 5 microns, those will fall to the ground. We shifted that in total agreement by all scientists and engineers that things clear up to 100 microns can actually float in the air and go much further than 6 feet. And then finally, we read a review looking at, and Shelley showed this image before, but the point was that it's not just SARS-2 that's airborne, it's all respiratory viruses. And that shouldn't really surprise people. Again, it's a head scratcher to think about, why are we surprised that something that is a respiratory infection is inhaled? But that's where we are. And so, what I think is, and I think I saw a question in the chat about, is there differences in modality, different types? We have touch surfaces, we have inhalation, and then spray that can just, you're close enough that the drops can splatter on you. And so, we're sure there's some, different ratio of those transmission pathways. They can vary depending on indoor conditions and indoor design. They also can vary, I'm sure, depending on the particular pathogen you're talking about. But basically, it works for flu, a lot of different secondary infections, RSV, measles, all of these have some airborne component. And so in addition to sort of writing these papers, we were involved, this was with Lindsay Marr. We did, I don't know how many different media appearances. I want to do a shout out to Dr. LaPook, who's the chief medical correspondent at CBS. He, from way early on, I think I had my first conversation with him on June 1st of 2020 after that perspective. And we just kept hammering. We did, I don't know how many shows talking about, it felt like we were broken records. I still feel like a broken record. But basically we made the point that this virus is airborne. And the whole message was, people would say, don't say that because you're going to scare people. And that is so far from the truth. Everyone that I've ever told that the virus is airborne and told them then how to protect themselves has been grateful. Nobody has been scared. Once you understand how it is spreading, you can protect yourself. And so, this is a very fixable problem. And I think that has been a big frustration for many of us. So as I mentioned, size plays a critical role in many different ways. A lot of times people will say, well, viruses are really tiny. They're like 10 nanometers, 20 nanometers. But they're not that way when they're floating in the air. Remember, they come out of your lungs. And so they have respiratory fluid that gets dried down on them after they're released. So, SARS-CoV-2 is probably about, 150 nanometers, 0.15 microns, something like that. It's much bigger than, just by itself. It can go all the way up to microns, depending on how much respiratory fluid encompasses it. But this size will determine how long it stays in the air. Part of aerosols can remain in the air for seconds. Big stuff can fall really quickly to hours. And so, how far it will go, how quickly it will drop to the ground, and where it will ultimately end up in your respiratory tract. That's kind of where we think the 5 micron might have originated. The smaller ones can go much deeper into your lungs. Aerosol particles can get into every organ in your body. As they get smaller and smaller, they can actually cross the blood-brain barrier and get up into your brain. And so, and finally, it affects how well you can remove them using PPE and masks and filters. And so, ultimately, size is really an important parameter. And one thing that we have really come to appreciate, at least for SARS-2, is that it's largely in the aerosols, it's in the smaller particles, and that's different than people believed historically for many, many decades. And so this slide I like to show because it's part of the history, I think, at this point. I use this, it was handed off to me by Lindsay Marr, and I showed it to Tony Fauci in a Zoom call in August of 2020. And his eyes, I just will never forget, his eyes got great big, but it basically shows the height something is sort of dropped and how far it will go. So the distance is on the x-axis. So this is for low indoor air velocity. So this gets into indoor design. Small, even a 5 micron particle can go, in this case, 100 meters at a low indoor air velocity, and a high indoor air velocity can go 400 meters. And so, again, this is just sort of assuming that things are allowed to sort of fall as a function of their as a function of their size, whereas we know in indoor environments, things can get suspended and held up in the air for even longer. And so this is just, again, an aerosol sort of coming out of somebody. If you cough or sneeze, that propels it even further. So he was shocked when he saw this. This was, an aha moment, I think. And after that, he started talking about aerosols and calling us aerosol activists, some of us, because I was kind of, I don't know how else to say it, I was kind of relentless. And he helped, actually, he read a lot of the papers that I wrote, and he actually kind of flipped. And again, for someone who thought forever that 5 microns was somehow this magical size, above which everything would just fall to the ground and don't worry about it, he really, sort of woke up to sort of how far things could actually go and became a proponent of aerosol transmission. So just linking, I still might not have convinced you that aerosolization from the ocean has anything to do with aerosolization from your lungs, but this is the vision of what, what we think of as sea spray production coming off the surface of the ocean. These are beautiful modeling simulations one of my colleagues, Romeo Amaro, has done. She really was working on SARS-CoV-2, and she's looked at respiratory aerosols. And it's shocking how similar the composition are of these two different systems. And again, you look at the surface of your lungs, the surface of the ocean, and backwards. And what you start to see is, they have incredible similarities, including the aerosolization process. So it's not as big of a stretch as you might think. And so one thing that, again, just sort of the background is to any aerosol scientist or any physicist who ever has studied aerosolization, including Lydia Morawska, who's been a warrior in this whole thing. Basically, one of the things that's been said throughout the pandemic is, we've learned so much. We didn't know this before. But I want to point out that we did. This is papers from Lydia's group dating back to 2009, looking at the size distribution of what comes out of you, humans, the aerosol producers themselves. And, it's not coughing and sneezing. It's just talking and singing. And the louder you get, the more vigorous you get, the bigger they are. But the point is, basically you can see that most of the things that are produced by humans and aerosols are produced in much higher abundance than droplets when you're speaking anyway. Again, coughing and sneezing is a different story. Kevin Fennelly actually wrote a really good review on this and said, he hadn't really found any direct evidence for large droplets as a mode of route of transmission. It just doesn't make, if they were droplets and they fell to the ground, that would require everyone to sort of touch the same surface or lick the same serving fork. It doesn't make sense. And I don't think that I can't think of any way that having droplet production alone would ever lead to a pandemic, especially like the one that we're honestly still in. So these were slides that were made for me. I just have to show them because they were made by Canadians. I got to know people all over the world who've helped me prepare talks and debates and all these different kinds of things. I did a debate with Dr. Fissman, with a very famous infection control person that has been mentioned. But the point is, basically you can get long and short range aerosol inhalation. This is the main way that things move around. These are the features of what we call super spreader events. One thing that has been noted is that if we could just eliminate super spreader events, where 10% of the people lead to like 80% of the infections, many times people say, well, I have been exposed 10 times and I've never got it, so it's not that contagious. But if you happen to be with one of these super spreaders, or in the conditions of that allows a super spreader to happen indoors, then that's where like everyone gets it. It's not just onesies and twosies. That's not what's driving it. It's this massive spread that happens. And so, bottom line, there's evidence. This is one of my favorites. This is a person who's singing, a chorist who's singing, has his back to the audience, and you see the color code for all the people that got infected. So one of my quotes I said before is, droplets don't do U-turns. They will just continue out. Aerosols will float like cigarette smoke, and they can bend back around. And that's the only way you can explain these infections. Same with these poor little ferrets. They put multiple bends. Droplets don't like bends, especially at angles. They will crash out, but only aerosols can lead to an infection from the bottom infected one to the top one. So there's all kinds of evidence that has accumulated. I can't even, I used to be able to keep track. I can't even keep track. The other thing that happened during the pandemic was there was a huge amount of shifting of the goalposts for what qualifies as airborne transmission. And one of the biggest things that people pointed out when we finally said, okay, we've proven it's in the air, they said it's not infectious. You haven't shown that it remains infectious. You might detect it coming out of a person. You might detect it in a hospital room, but it's not infectious. Well, in fact, Lednicki and others showed with special sampling, because air sampling is pretty harsh on bioaerosols, it's been shown now probably, I don't know, at least 10 times that after you collect the virus in the air, SARS-2, it actually does indeed remain infectious. And this is really important because I want to point out that something like measles, which people have no problem calling infectious, still has never been shown to be infectious after being collected in the air, but SARS-2 has. So, SARS has really, I don't know, just been proven, I think, beyond all doubt that it is being transferred mostly through the air. This is a slide that I worked on, or I borrowed from Lindsay Marr. It was in this great paper, and basically it just shows our current understanding. There's short range aerosol inhalation. These are the big droplets I mentioned, those fall quickly to the ground, and then long range aerosol inhalation. And these are sort of the factors that have led largely to the spread of this virus. So there's been a huge debate. Is it mostly long range? Is it mostly short range? Again, it really depends on the conditions. This is a classroom, which they went and looked at the effects of masks and no masks, and were able to show that, it's the long range airborne or long range aerosols that lead to the most transmission. This just came out in 2023. So one thing you can do, again, from a design or modeling perspective is, there's these things called CFD simulations, and there's computer simulations where you can beautifully simulate how air will flow in a room given obstacles and ventilation conditions and people and all that. And so I just want to show one example. You can take an infected student, and this is large, one way you can actually get at how many, Shelley had mentioned, how many air changes per hour you need. Again, it depends how many windows you have, how many windows you can actually open. And so, I just want to point out that there are really amazing modeling exercises, but you can also just go in the room and measure using CO2, the ventilation levels, or using particle measurements, the amount of aerosols suspended in the air, and how these change as you begin to implement protective measures. So I just want to, again, point out, I think one of the biggest things is once you acknowledge it's airborne, then people begin to understand, and this is what I've just wanted to happen we've all wanted to happen for 4 years, everything starts to make sense. Like light bulb on, what leads to the most spread? What is the riskiest thing I can do? And it's crowded indoor locations where people aren't wearing masks and are yelling, basically, is what it comes down to. So if people are quiet, you can see it's green, it's not as risky. Think the risk drops if everybody's wearing masks, but the worst area is over here, which is, high occupancy, poorly ventilated, and people shouting. And so the other thing I want to mention, and again, it's confined spaces. So restaurants, bars where people don't tend to wear masks, cars, buses, school buses are terrible. Elevators are terrible. Public restrooms, as I'll show towards the end, are awful. The bottom line is that, the virus can remain in the room even after everybody's gone. it can stick around for hours in a poorly ventilated space. So a lot of times people will walk into a corridor or walk into a break room and nobody else is there. And what do? Even if they were wearing a good respirator, they take it off, right? But the virus can still be present, especially in public spaces. So layer protection, this is the Swiss cheese model. It was really, it's been a lot of people have shown this in various versions. I don't even know if this is the latest, but the point is no one measure protects you in every way. You have the more layers of Swiss cheese that you stack, the safer you will become. And so, but Shelley focused a lot on the ventilation side of things. And we've known ventilation is important. This is Florence Nightingale. She always was opening the windows. They were measuring CO2 levels back then. And so, basically, we've known ventilation is important. I have been a big proponent of filtration, supplemental filtration, for a variety of reasons, just adding, again, that extra layer. And there's been a DIY solution that has been amazing to be part of, where people can build your own filtration device. Think about it. We filter our water before we drink it so that we don't drink pathogens. We drink about 2 liters of water a day. We breathe 11,000 liters of air a day, and we're okay not filtering out the pathogens. It's the #1 way that we're exposed to pathogens. And so, the first people are just like, okay, they're cheap. You can build one for maybe, depends on where you live, $70 versus a HEPA that could be $800. But not only are they cheaper by a factor of 10, they outperform HEPAs. And so, and you can make them kind of cute. This is a minion in a classroom, which I think is cute. This is Shivan, who is in Canada, who has been leading the charge as an activist for cleaning indoor air. And so once you start adding these, each one that you add to a room, you can actually build up in terms of number of air changes per hour. And I want to do a shout out to the clean air crew, who worked really hard to sort of assemble all of this information that anyone can find, and it really helps make it doable for anyone. Kids, as I say, everyone can build these. So, low cost solutions. I worked with San Diego Unified, which is a very large school district. We put in measures to measure, in this case, this is CO2. So again, the amount of ventilation. We also had particle measurements, and we also used it as an opportunity to educate the public by putting these little QR codes so people could understand why these things were in their classrooms. And we also did quite a bit at UC San Diego. This is one of the builds we did where we put them in all the lecture halls. Also, with Marina Creed, we took one to the White House. So we basically just tried to get the information out as far and wide as possible. One thing I want to just mention as I'm getting towards the end is that speaking of design, I made this slide up just for this, is that, first of all, there's really great resources, Clean Air Crew, and now there is the Corsi Rosenthal Foundation, which is Rich Corsi and Jim Rosenthal have put together this incredible effort to help more people and to get these kinds of things out. One of the big movements recently has been designing better looking Corsi Rosenthal boxes, but not just better looking. You can start thinking about integration. I've been thinking, what about a coffee table? What about a wall? I mean, there's ways that this is where architects and people with building design, they should just become part of the structure, in my perspective. I mean, they kind of are in the HVAC systems, but HVAC systems alone aren't enough during the spread of disease. So winter and times where COVID is taking off. And so quiet is big. So they are using computer fans, pretty smart. Those box fans can be a little loud, but you're supposed to run them on low. People don't always know that. Inexpensive and high performance. But the one thing I want to say also, I think one of the biggest things I really love about filtration is that it also removes other things like pollen and allergens, and other people have a lot of respiratory issues. They live, as Shelley mentioned, some people live in horrible places where they're breathing the worst air. This will remove those things as well. And I would say it's something that gives it an advantage over UV in that you're physically removing these pollutants that people are inhaling. So one other plug for ventilation is it also improves cognitive performance in work, schools, wherever you are. Increased ventilation lowers CO2. People produce CO2 when they breathe. This is a simple CO2 monitor that many of us have. And you can see that the test scores drop as CO2 levels rise. And this is the average level that have been measured. It's around 2,000 in schools, which is terrible. Outdoors is about 400. All right, so proper public health messaging is absolutely key. I took this picture on October 12th and in our hospital, which is, I say this with a wince, because it's saying that hand hygiene is the #1 way to prevent the spread of infections. For an airborne virus, that's not. I mean, it's important to wash your hands, don't get me wrong, but to say that it is the #1 way is, where the public, I understand why the public is confused. We really need to work on getting out the right messaging. Spread through services was sort of dismissed early on by the CDC. And so, having access to the right masks, things that, at least KN95, some people say it has to be an N95, test fitted, but not these little baggy blue masks, as we call them, the surgical masks, which were designed for droplets and do not work for aerosols. So better public health messaging. I like this hospital better. They have air filtration above the sanitizer with signs. And so, we can start thinking about, ways to reduce not just SARS-CoV-2, but all viruses. Actually, things that create secondary infections in healthcare settings also have been shown to be removed with proper air filtration. So I just want to put a plug in. Lindsay Marr and John LePuc are going to be on TV on Sunday on 60 Minutes. This is a big moment for the airborne crew. We're all so excited to see this. John has just been the leader, I think, in the media. Lindsay she's been studying this longer than, most of us that came into this, and has been very embracing of us, me, asking her lots and lots of questions, but it's made it to 60 Minutes. So it's gotten to prime time. So we're all excited to see this, and it'll be on Sunday night. Finally, if we give the public clear guidance on how they're getting infected, I've said this since I said it to John LePuc on June 1st of 2020. It is a fixable problem, and it teaches you what to do and what not to do. And it also makes sense. You don't have to memorize where you're going to be safe or not. It's just logical. It's just pure logic. And the public does understand it when you explain it. And yes, public restrooms are a great source of bubbles and aerosolization, unfortunately. And they don't have lids. So that's something from a design, any sort of effect, we should be thinking about that. I don't ever go in a public restroom without a mask. So I'm going to stop there. And I thank you again for the invitation to be here. And I look forward to engaging in conversation.
Thank you, Dr. Prather, for sharing the significant research with the audience. I would now like to invite Mr. Gabriel Gianodoli to join us for the second presentation. So Mr. Gianodoli is a designer working on news stories. His work combines art direction, design, and programming to create interactive experiences. He was named the world's best designer by the Society for News Design in 2021. Mr. Gianodoli has worked on multiple interactive projects, including the Close Read and Pattern Recognition series and the effort to track the coronavirus. He previously worked at the Wall Street Journal and holds a master's degree in design and technology from Parsons School of Design. And I'll pass it over to you, Gabrielle. Thank you.
Thank you, Alice. Thank you, Dr. Prater, too, for the incredible talk. I think a lot of what I'm going to show will overlap with what you just said. I think we're trying to reassemble the history of the year 2020 with all that the CDC was saying. Let me just see if I can share my screen. Oh, I might need to restart Zoom just a second. Be back in less than a second.
Hi, everyone. We're just going to wait for Gabrielle to join us back again. So just give us a minute while he rejoins and we can get him set up again. So thank you for your patience. So just give us another minute or so and we'll continue. Thank you.
I think now this should be enabled. Okay. Can you guys see my screen?
Yep.
So let me see. Is the band from Zoom over the screen? Should I
Nope.
try to move it? It's not?
No, it's not.
Oh, okay, this is great. So let's start presenting. Okay. So like Alice said, I work at the New York Times. I'm an assistant editor for digital news design. I'm gonna explain what digital news design means in a bit, but for now, I'm gonna go over a real quick intro to myself. I started working in editorial design, which is like design for news, back in 2006, more or less. I was working in magazines at the time. These are some of the spreads that I did at the time, and these things were called infographics, and some people started to call them data visualization or charts. But in general, that's when I started to combine coding and design, which is my main background, to produce these data-oriented pieces. These were mainly for magazines. Then I worked at this studio called the Office for Creative Research, which was led by a Canadian, Jared Tharp. I don't know if anyone here is familiar with him. It was a data viz oriented studio as well. We did a lot of physical installations, as you guys can see. And after that, I worked at the Wall Street Journal in the graphics department. These are a few of the pieces that I did there. Some about the divide in the Republican Party at the time. Another one about the outcome of the 2008 financial crisis in the US. And then in 2018, I joined the New York Times in the digital news design team, which does custom presentations of digital stories. So what does that mean? 99% of the New York Times stories that you guys see go through the CMS, the content management system. So they don't involve a designer doing anything custom for them. The reporter, alongside an editor, sometimes a photo editor, is able to put together that story and it gets published. A few other stories, they go through this special CMS, if you will, like a system that we built internally to do custom stories. Some of the examples are this close read series. Maybe you guys have seen that before. We analyze an art piece, we zoom into details, and the art critic Jason Farago talks about, say, a painting or a photograph, show also inspirations that that piece is related to. Another example, the series called Pattern Recognition, where we show multiple instances of something. In that case was Meryl Streep wearing glasses, like how in different films, just trying to convey a different message through the glasses. But we talked about other things as well. And all of these four that I'm showing now are from the culture desk, which was attached to until maybe around 2020. But we also do a lot of works with the graphics desk. which is the graph, the desk that gets involved when we are dealing with a map or a chart or a 3D visualization, which is the things that we're going to focus on today. So some examples of those are on the top left. This was a comprehensive collection of Black Lives Matter protests that happened in the US from May to June in 2020. The social distancing piece that I'm going to go over that one in more detail. This masks piece that I'll talk a little bit about that one too. And on the bottom right, it's an older one about the elections in 2020, where there's a combination of the politics and the culture desk. Actually, we combined an analysis of the playlists that the candidates were using in their campaigns and tried to gather some information as to how the songs that they used related to the personalities and the messaging that they were trying to convey. So the piece that I'll go over today in more detail is this one. I'm going to scroll through it and then go back to some kind of making of. So like Dr. Prater was saying, the main focus of the COVID prevention in the beginning was the thing about washing hands. We still have a lot of signs inside the New York Times too. I should have taken a picture, like, don't forget to wash your hands. You need at least, I don't know, like 24 seconds. And so there was a lot of confusion because then the messaging about social distancing it started to be conveyed. And I'm going to go back to the timeline too, but just to give you a brief context of when this piece was produced. This piece started in mid-March and it was published in mid-April. So it basically goes through what the main recommendation at the time was, the thing about 6 feet, but shows that we don't go over the precise sizes of the particles that we're talking about. We simply split them into larger droplets and smaller droplets, and then show that, like the CDC and the World Health Organization were saying, the large droplets, yes, they fall to the ground closer to 6 feet, but the smaller particles or aerosols can linger in the air for a longer time and spread further than 6 feet, which in countries that use metric, I think was translated as 2 meters. So in this 3D simulation, we show those particles and you see that they will start spreading further and further from the 6 feet ring there. And they can get to even 26 feet, depending on the intensity of the emission there. I think 26 feet, it's from a sneeze, could be 16 feet from a cup. And this is a simulation based on indoors and no ventilation. And we also talked about how even if you don't cough or sneeze, simply by talking for a long time, viral particles can linger in the air. And it ends with a call for people to use masks too. That was when the conversation around masks was just starting. I think when we started to do the piece, basically people were just calling for social distancing, and by the end, people were already talking about masks. Let me see if I can go back to full screen. Yes. So putting together that timeline again, like Dr. Prater was saying, this is one of the first charts that the New York Times published about COVID. This is March 3, and as you can see, like the cases in the US were still below 20 cases a day. And soon that would evolve to what we internally called the COVID tracker, which is a project I worked on for a year as well. Where, as you can see, like the cases in the world were reaching hundreds of thousands a day. And you guys may be familiar with this page, with this templates, but we had a page for the world, a page for the US, pages for a few other countries too. And by the end, we had pages for each state in the US and for each county in the US, for each city. But going back to the timeline, the Times published this piece in March 11. And as you can see, the wording we're using here is still to talk about the droplets. We say influenza, whooping cough, and most likely COVID-19, according to the World Health Organization, are transmitted via the large droplet. So by that time, there were a lot of researchers questioning that orientation by the World Health Organization and the CDC. And as we were reporting, this is a story, this is a small detail in a larger story. So the focus of that piece wasn't this chart that you guys are seeing. So that chart didn't go into much detail about the transmission, but it also says that other diseases, which are called airborne, could spread further. And then around March 15, at least in the US, I think this is specific to New York actually, restaurants are closing. This is US wide actually. The CDC was calling for cities to close restaurants, to don't allow gatherings of more than 50 people, and to practice social distancing. So which was just 6 feet apart. So that piece, one person who worked on that, the other piece that I was showing was Yulia Parchina-Kotas. She's participating in this symposium too. She's going to talk tomorrow. So because she worked on that piece, she started to suggest a new piece that looked deeper into the viral transmission via droplets, not only large droplets, but also small droplets. She interviewed a few researchers for the other piece, and all of them said, well, the CDC can't be sure that it's only through large droplets, and we have studies showing how viral transmission through aerosols happens. So we approached this new piece with this caution there that we were going to talk about viral transmission through aerosols, but not necessarily go into the theoretical discussion of what is airborne, what is borne through air, like Dr. Prater was showing too, like the Wired published that other piece about that too. So this is one of the first sketches that Yulia did, showing a person and the 6 feet line and how the large droplets fall and how the aerosols can spread further. I started to gather some visual inspiration too for the colors and the arrows and the visual elements that we were going to use on the piece. I have a folder with a lot of visual material from early 2000s and things that I save on my hard drive. So these are really old. I'm sorry for not having attribution on them. But those are old infographics and also concept art sometimes. It's things that I use to study color and contrast and how to show things and apply those things to graphics that we might be doing currently. These are a few images from research papers that Yulia was using on her reporting. They show photographic images of the particles being released and the behavior of the particles if you're wearing a mask. And the main challenge here was that we had decided on this 3D simulation from the beginning because there was a lot of discussion on how how do these things work indoors versus outdoors? Is it effective? What's the correct distance and all of that? We knew that if we got closer to reality, if we had a 3D simulation instead of just a 2D drawing, we could convey a better message to people. It could be clearer in what we would be informing people. So these are some of the studies that we did based on the 2D images from those research papers. And they're not very satisfying at this point because you guys can't see because it's a silhouette, but if you put that in 3D, it's very clear that you're dealing with a 2D language and applying it to 3D. So it wasn't very effective at that point. So Julia also tried to use some particles that she programmed in this 3D software, but they're just falling through some mechanics that she applied to it. This is not based on any real simulation, and we didn't want to use that because I don't know who among you here remember there was this viral video of runners and how the droplets could also spread when they're running and they could contaminate each other. This study was then debunked as false. We didn't want to cause panic on people by using something that wasn't completely scientific accurate. So the breakthrough for us was when another reporter working with us, Badal Sajid, he found this video on Japanese TV from researchers from the University of Kyoto, and it showed a simulation of particles traveling through space in indoors in a non-ventilated space. So even though there's a note about the study not being peer-reviewed in the end of the story that we published too, but the thing was, what this study was showing was backed by all of the researchers that we talked to when reporting on the story. So the breakthrough for us wasn't that we found these researchers in Japan who had a completely different message. It's quite the opposite. We found researchers who had 3D data that allowed us to visualize what the researchers here were talking about, the thing about spreading for more than 6 feet. So this was a breakthrough for us because then we could get this data with these dots that were positioned in 3D. We had X, Y, Z for each of the particles there, and also had the size for each particle, which is what this chart here is showing through the color scale. And we could then use that, import that into the 3D software that Yude was using, and then finally apply that to the story. So this is one of the first videos using that. And this is just one snapshot. So the data, it's actually 5D, if you will. It's the 3D of the dimensions plus the size, and it also has the time for each particle. So we started to experiment with that. Again, this is not incorrect here, but it's not corrected to the size of the particles. They all look the same size, so it's a very scary image if you had published it as is. Also, the person is very close to the 6 feet line, and we wanted to avoid also being conveying a message that caused panic on people. You know, we wanted to show exactly how we worked, but without causing panic. So this is closer to where we landed. We started to experiment with the colors for visualizing the sizes of the particles. And, you know, even though we were using red as the accent color or emphasis color on most of the visualization, red applied to the droplets here just looked like blood. So it was a bad choice. We tried other colors too. We even tried a scale of colors with showing the grading of the sizes like you had on this visualization here. But again, the thing is, a scientific visualization has a different audience, right? Researchers are interested in all that distribution of sizes, but for a general audience here, we were mainly talking about the difference between large droplets that fall to the ground and aerosols that spread further than 6 feet. So we didn't need to show more than that. So going back to how the final one looked, we ended up using this blue color for the large ones and then white for the small ones. Another small thing here is we try as much as possible to diminish the amount of cognitive work that the audience needs to do. So instead of doing a key with colors someplace else on the screen, we highlighted on the text. Blue is large droplets, white is smaller droplets, and this is all that people need to understand when they see this. And going back to this. We also did a lot of studies on how to show the 6 feet distance, the focus on the ring itself versus the focus on the area, which seemed more appropriate. It's what we used, the one on the right. And like I said, when we approached the end of the story, I think it was towards mid-March, the conversations were already shifting to talk more about the use of masks and how masks are more important than, I mean, in conjunction, like the layered system that Dr. Prater was showing, in conjunction with social distancing and all the other practices, masks are very effective in preventing COVID-19. The piece also had this AR component. This is a promo video that we shot. We went to supermarkets, we walked on the street, and if you had the New York Times app at the time, you could install this AR piece on your phone, and then you could see and compare like the 6 feet distance, how that measures in real life. It was interesting to do this. We saw that it was quite impossible to keep a 6 feet distance in New York that is so crowded. Even with the social distancing guidelines in place, there were a lot of people in the supermarkets or on the street too. The Times published a lot of other pieces on particles, COVID, and ventilation. This is one of them. This shows how a cough or sneeze can spread in an airplane. The bottom line here is the ventilation system in an airplane. It's actually pretty good. It renovates the air at a very high rate, and it contains the particles to a few number of rows, so it doesn't spread to the whole plane. The Times also did this piece on the ventilation in the subway. This is already August. And as you can see, these pieces are not theoretical exercises on particles and ventilation. They were all responding to topics that were current at the time. People started to ask, well, can I travel? Is it okay to go on an airplane if I try to social distance before and if I use a mask? By August, some people were going back to work, had questions about the subway. And then by February, people were discussing if schools would reopen or not. And the simulation here showed how ventilation in schools worked. And each of these pieces uses a slightly different representation for particles and airflow. So as you can see, there's not an answer an objective answer for how to represent these things. And they go through several different visualizations as well. Go back to the slides. And in October, we published this other piece about masks. We're near the end here, so I'll be really quick with this one. Also because Yulia is going to talk about this one in more detail tomorrow. But like I said, this was a shifting conversation throughout 2020. And by October, people were already convinced about the masks, but had questions about N95 versus a cotton mask. So we started to look into how masks worked. A lot of the visualizations on that showed this 2D diagram that was, very abstract for people. We looked into scientific papers on how the mesh on masks look like. And again, the decision here of using 3D was, let's make something as close to reality as possible so we can convey a clear message on this and show people how this works. These are some of the initial studies showing arrows for the flow of the particles, but we ended up actually moving the particles through the piece. And we built this UI, these sliders on the right there, so we could tweak the colors of the particles, the colors of the background, and everything that you're seeing there. We could tweak it on the page, so we got to the final colors of the visualization too. It ended up looking like this. Let me see if I can scroll through. It starts with this group of people. We show the difference in the mesh of the two masks. It zooms into the cotton mask now. Briefly shows 3 groups of sizes. Again, we don't go into a full distribution of sizes there. It's something simplified so people can understand. Large particles are easily trapped by the blue mask, the cotton mask. But then N95s have the electrostatic aspect to them that attracts the particle and then traps them on the mesh. And this was it. Usually we'll talk about this piece more tomorrow. And thank you so much for listening. Hope to answer your questions too. Thank you very much.
Thank you, Mr. Gianardoli, for sharing those compelling graphics with the audience. Now I'd like to ask Dr. Prather to join again so we can begin the Q&A session. And so just a reminder for the audience, please share your questions for Dr. Prather and Mr. Gianardoli through the Zoom Q&A function. I think we already have some questions coming in. So let me just pull that up. There's a question for Dr. Prather. The droplets, if they fall to a surface and then someone were to touch it without cleaning it, they're wondering if that's still a way of getting infected. I guess what's the difference between that way of transmission compared to air particles that travel further?
Yeah, I mean, that's a great question. And I think what it depends on a couple of things. One is just, and I think was in the question, is sort of the ratio of the particles that will float versus those that will fall. And for SARS-CoV-2, there's just not that as many droplets unless someone is sneezing and coughing, which is starting to become more common. There's another thing that happens though. It's not just the ratio, like that more will float than fall, which is pretty true. But the other thing that really matters is if something does fall on a surface, is how long it can stick around and remain infectious. So it's, there's been studies for for SARS-CoV-2. There haven't been that many studies, but there was one on surfaces where people looked at how long it remains infectious. And I think it was, I want to say it was like up to 6 hours or something. They only looked up that long, 4 to 6 hours, something like that. So yeah, that's really the question is once this virus when it seems to be, sort of lose its infectiousness after it's sitting on a surface more than some others. It's due in large part to its structure, actually. But yeah, that's kind of the long answer is yes, you could still get infected, but for one, it's like there have to be a lot more droplets and they have to remain infectious.
Okay, thank you, Dr. Prather, for the information. And a question for Gabrielle. So much of the final work is graphic. How do you decide how much text should be included? It seems like very little is needed because the graphic contains so much information.
Yes, especially when we are doing these kind of stories that we call internally scrolly telling, when you have something that is scroll triggered and the text is scrolling over, we are very conscious of the amount of text that we need, because all of those screenshots that I showed were desktop because I was doing this presentation on desktop. But if you look at that on mobile, our main and practical concern is the paragraph size, does it fit a mobile viewport without overlapping with the whole graphic that you're trying to show, So there is that. We really go over many iterations of text so that the paragraphs are short, and the story in itself, we try not to make it super long. And also these are different. It's not like the whole New York Times coverage on COVID transmission is that story, right? There were longer stories there were stories showing interviews with other researchers, so that's a very focused piece. So that's why we can afford to be short on that one.
Got it. That's an important note to think about, I guess, how the information is delivered. And I guess in a related question for Dr. Prather, what are some effective ways of using science to combat misinformation about disease transmission and appropriate measures?
Leave it to Dr. Siegel to ask me a zinger. He's one of my indoor air heroes. Yeah, so I mean, as far as I ask myself that question all the time. It's kind of like, how do you get through to people? And Gabriel did a great job of showing visualization as one way. I've tried to explain things so many different ways, so many times. Sometimes you get through, a lot of times you don't. And as Dr. Fissman talked about, sometimes people, for whatever reason, just almost don't want to know. And so a lot of the problems that, and I don't know if we'll get into these, but it's something I think about a lot. And one of the places we're sort of settling down is, I used to think that the reason people were making the wrong choices and getting infected and it was spreading so fast, I used to think it's because I just wasn't explaining it well enough. But I don't think that's the case anymore. I think that it's human choices. And why do humans make the choices that they make? And so this is a huge, it's like climate change denial. It's turned into a huge we need social scientists. We talk about another field. We need social scientists. We also need people like with different backgrounds. I see where the designer community could play a role, is almost like, that's why I mentioned integration. Just making clean air a part of the room so it almost doesn't pop out would maybe make some people happier. It's how do people's brains work? When there's a deadly virus that gets into every organ in your body, and why are people like, now we're just acting like it's all over, and it's not. And so how do we make it part of our everyday lives to clean our air just like we clean our water? And that's policy. It's mandates, which again, people hate that word. Mandates elicits a visceral response from many people because it takes away our freedom to get sick and die. But yeah, it's just, sorry, that was my dry frustration coming out. But yeah, so Jeff, I mean, I wish I knew the answer because I, have been banging my head as others have into figuring it out. But I think I can sleep at night not wasting time trying to figure out a different way I could explain things better, a different figure I could use, a different study we could do, right? It's not the science. It's like, just like climate, we know what we need to do, yet the majority of people are choosing not to, and many people are confusing that messaging. Many of the same people who confuse it over climate and tobacco smoke are throwing in missed messaging. So all we can do is just keep trying to get my mode is I can do so much, and I'm just going to keep sending out the correct message over and over and over, and won't be silenced. So yeah, it's a tough one.
I love that there's an overarching theme of our symposium of how important collaboration is across disciplines. We have a comment for Gabrielle. Thank you for sharing your graphics. It's interesting to get a glimpse into the thought and details that went into creating the graphics. And then also a question, were there other stories like the ones you showed that were not published that you wanted to see through?
No, I think we did publish a lot of things. I tried to collect as many as possible as I remember. That's why I went after the plane one, the subway one, school. I'm pretty sure I missed something, but I think we did a good job of covering that topic. The COVID tracker in itself was this ongoing story, right? It's unprecedented in the history of the times. It was almost like an app. We met daily to discuss it, and we changed many of those charts that we showed to show, say, anomalies when states weren't collecting the right data, to cover different mandates. So that was a multi-day, year-long story, and I think that was like the main story that we worked on for a long time, and it covered many things in COVID.
Got it. We also have a great comment from the audience member. It's not information deficit problem, it's a getting the message in problem. Just wanted to highlight that. And I think this might be the last question as we wrap up. But for both of you, are there any priorities or important factors that interior designers should consider in the range of work they do? From both of your expertise, whether that's designing graphics in the interior spaces or coordinating ventilation and the such.
If I may, I found it interesting, the question about text. I don't know if the person was trying to get to that, but there is really this difference between a poster that has 200 words versus a poster that shows like 5 pictures and a few words in that. Not saying that scientific information needs to be dumbed down for people to understand. Quite the opposite. I think that seeing things visually can convey a lot of precision, and it can help people understand things faster than if they need to read, I think, something that it's text only.
Yeah, I'll just quickly say, I think that, just the concept of making clean air part of the space, because people, again, I love Corsi Rosenthal boxes. My heart gets warm when I see them in the wild, because I know what they're doing. They're saving lives. But a lot of people don't. They'll say they're ugly. Sorry, Jim and Rich, but they'll say they're ugly. They have duct tape, they're noisy, even if they don't have to be. And so to me, like what I've been thinking of I personally am thinking of ways to, design to get things to be, also designed just to get them to people. So they're not some big cumbersome thing, but again, putting them as walls, putting them as part of structures, like making them part of art would be, and I've seen some that are getting pretty close to art, but making them part of the structure, just part of your daily lives, where they just kind of are, they're an automatic thing that you expect to see there, like a seatbelt, putting on your seatbelt. It just should just be automatic that if you don't feel clean air, you don't feel well, there's something off. But how can we get things to be that automatic? And I think a lot of it some of it could be public appeal.
Well, once again, thank you, Dr. Prather and Mr. Gianardelli, for sharing your insights and answering questions today. That'll help interior designers gain perspective for creating healthier interiors, especially for our Sunday workshop. So this closes session 2 of the symposium, and I'll welcome Lois back to the screen for closing remarks.
Great. Thank you so much, Kimberly, Gabriel, and Alice for this incredible session. I think has us thinking in a whole different level at this point now. There's been so much information that's come in just from all these different perspectives. And in this last session, it's so nice to see how some of the same graphics even show up from completely different disciplines, which I think is so incredible and does go back to this idea of how we can be interdisciplinary and learn from one another and bring that information from one area of knowledge into another. I know for interior design, all of this keeps kind of circling around and through it, just like those incredible graphics that both Kimberly and Gabrielle showed. So this means we're wrapping up our day one of the symposium. There's been lots of different questions coming in, some about whether or not this will be the recordings will be provided. And so that's one of the things that we'll be working on in the next couple of weeks is getting them onto a platform to share this content, because it's really important to have it out there so everyone can learn from all this expertise. So please, keep checking back on the Healthy Interior Design website for that information. But I know this has just been an incredibly rich set of presentations and so much content that I know the team here is going to be coordinating to keep continuing and building a set of guidelines for interior designers that we can also use in the workshop that's coming up. I just once again want to thank everyone that has been involved presenting today and moderating, when I know it's a lot to kind of summarize your work in 30 minutes when it's a life's work that you're doing. And it's so important to every day, everything that we're doing throughout our lives, and wonderful to see how this is getting out there. So we're happy to keep those messages continuing. So thank you, everyone. Tomorrow we start at 9 a.m. If you heard me give the introduction this morning, you can sign in at 9:15 and not have to hear the same introduction. But we'll be back again tomorrow for 4 sessions. Today was 2, tomorrow is 4, with a lunch break in the middle. So you'll have a little bit of break and hopefully a little bit of a more break between the sessions too. But when there's so much interesting content, there's no sense in stopping it. It's more important to have this content. So thank you, everyone, and look forward to seeing you tomorrow. Thank you.